Hello, thank you so much for coming to see my poster. Today we're going to be talking about the molecular correlates of training that contribute to memory formation after learning that food is inedible in Aplysia. This research was done over the summer of 2021 in the Brain Sciences Center at Bar Ilan University in Israel. In Dr. Sussmein's lab, I worked under Dr. Schnitzer, postdoctorate, who's part of the lab, and I worked alongside Sajida Mahamid and Meital Melol, both students at Bar Ilan University. So, like in the title, we were very interested in seeing the molecular correlates of memory formation, and we use the animal Aplysia as our animal model. And you may ask, why is it that we use a marine sea slug to better understand memory formation? It seems like an unlikely animal. And the answer to that is it's actually a great animal model for understanding memory formation for a couple of reasons. One of those reasons is that it actually forms memories, and there's ways of developing different kinds of learning paradigms where you can see them actually learning and forming memories clearly. Another reason is that they have neurons, which is great because if you want to understand anything to do with human learning, we use neurons, so their neurons function very similar to ours. Another thing is that their neurons are quite large in comparison to most other neurons. A singular neuron can actually be seen with the naked eye if you have good eyesight. And their neural circuitry is actually relatively simple. So instead of having one big clump of a brain, they actually have different ganglia, which are groups of cells that are responsible for different activities. So because of the fact that they have, the human brain has tens of billions of neurons, the Aplysia brain has tens of thousands of neurons. So it's easier to work with, which is another reason why it's great. So again, we're working with Aplysia, and we want to understand what are the molecular correlates of learning and memory formation. So we made the decision to look at CEBP and CREB1 because those are transcription factors that had been seen to be involved in memory formation. It's pretty well known in the scientific literature that it's pretty well established that they are associated with memory. So we went with those. In Aplysia, however, CREB1 is further processed to produce CREB1 alpha and beta. So we looked at those three, CBP, CREB1 alpha, and CREB1 beta. And these transcription factors turn on other genes that can produce mRNA to be transported, just a little background, from the nucleus to the synapses to produce long-term changes, such as potentially behavioral changes, or perhaps increased connections between different cells when forming memories. But we don't know. We want to see if these different transcription factors, if the genes that produce them will increase in expression when these animals are forming memories. So in order to do that, we had to set up a task that would have these animals form memories. So if you look at figure 5 over here on the methodology section, you can see the little diagram. We basically had a tank with a little divider in between with two different Aplysias. The reason why we had two in there, just a side note, is because when they're alone, they don't sense the pheromones of another animal, and they get very stressed because they were looking for a mate. So because of that, we have two in the same tank so that they can learn properly and are also interested in the food. So I just mentioned food. The task that we had is we took a food that they would normally eat, ova, a kind of seaweed, and we wrapped it in green mesh, so it was the same color as the seaweed, and we would feed it to them. So we would stimulate their lips by putting it by their mouth. And in the full training, we would allow them to bite down on it. They would try to swallow it, but because of the fact that it was wrapped in mesh, they weren't able to successfully swallow it and have it actually enter their gut. So that would be a full training, 15 minutes of that, where they would continuously try to eat the food. And then we also did a different kind of treatment, which was lip stimulation, where it was the same exact thing. Just before they would bite down on it, we would remove the food. So it just involved us putting the food by their mouth, again, for 15 minutes in our specific project. So now that you know how that works, I can talk to you about previous research that had been done on this topic. So here. All right, so first we have to show that this kind of task actually forms memories. And if you look here, we have a diagram in figure 1 showing that animals indeed did form memories after receiving 15 minutes of training. So just like I said, 15 minutes of giving them inedible food and them not being able to eat it. So if you look over here, A, B, and C, they're all very similar. They all show animals that on this left diagram, how much time it took for them to stop responding to the food the first time that they were introduced to this inedible food. So you can see in A, they stopped responding, on average around like 23 minutes. B, they stopped being interested in around like 30 minutes. C, they stopped being interested like 15, 17 minutes. But then 24 hours later, they did the same exact thing to the same animals where they gave them the same kind of inedible food, and the time needed to stop being interested in the food decreased significantly. So if you look here, the bar to the right is showing 24 hours later, the time it took for them to stop being responsive to the food. And because it decreased significantly, this shows that there was indeed significant memory formed from this learning task. So we know that we can use it. Okay, so what previous research had been done in my lab on the different molecular correlates? So on CEBP, which is one of the transcription factors that I spoke about earlier, they saw that its expression did indeed increase in the buccal ganglia when the animal experienced that 15 minutes of full training. However, it did not increase with just lip stimulation, and this was all in the buccal ganglia. But when they looked at the cerebral ganglia, they found that expression did not increase in training or with lip stimulation. So that's interesting to note because the buccal ganglia is actually responsible for movement and motor sensory stuff. So it's interesting to note just because you would think that maybe the cerebral ganglia would have an increase, but no, there seems to be an increase in the buccal ganglia. And then if you look over here, you have a graph, figure 3 and figure 4. Both, one is in the buccal ganglia and one is in the cerebral ganglia. This is from research that was trying to see if there was an involvement of nitric oxide in forming memory. So the training sessions were only 3 minutes in conjunction with different injections of salt water or nitric oxide. So it's not exactly the same kind of setup that we had because we were doing 15 minutes of training and lip stimulation, but it does give some sort of a story about increases in expression. But again, we needed to do further research based off of the study, which is what I did this past summer. So back to this, figure 3 and 4 show different kinds of training that these animals underwent. And this is in the buccal ganglia. So in the buccal ganglia, they saw that there was an increase in most of the different genes when training was done. But if you look here, you can see that with CEBP, there really wasn't necessarily so much of an increase in the cerebral ganglia. So you can see here This red bar is pretty low in comparison to the other ones. And then if you look at lip stimulation, in the buccal ganglia, you'll see that there's a very small increase, again, in CEBP, even when there was an injection that was supposed to help with memory. But if you look at CREB1, CREB1 alpha and CREB1 beta, you can see that there's more of a significant increase. Same thing with the cerebral here. You see how low CBP is in comparison to the others. Okay. Also, so what do we get from this? We get from this that there are increases in gene expression in CREB1 alpha and CREB1 beta, and also in CBP and the cerebral ganglion with just a lip stimulation. So this gives us some sort of a hint that there is something going on with increases in gene expression when they receive some sort of training, not exactly the one that we're looking at. Okay. Another thing to note that was important with these other two studies, one study was in figure 1 and the other study was one study was figure 2 and then the other study was 3 and 4. Is that in the first study in figure 2, the tissues were collected 15 minutes after the training. And in the second study, the tissues were collected 2 hours after the training. And this is something that's important to note because there may be peaks in gene expression earlier on or later on, and then they can go back down. So it's important to control for that, not knowing when the peaks in gene expression are. So in our study that we did this past summer is we collected tissues from the animals that underwent some sort of treatment, both 15 minutes and also 2 hours after they underwent treatment. Okay, so now that we know the background and what's different with ours, we can go into what we did. Just a quick methodology. So like I said, we had the training, we had the lip stimulation, and then we sacrificed the animals and dissected the buccal and cerebral ganglia, both 15 minutes and 20 minutes after the treatment. So just to go over it, we had 5 groups. We had short train, which is 15 minutes after a full training. Long train, 2 hours. The tissues were collected after a full 15 minutes of training. Short lip, so 15 minutes after lip stimulation, and then long lip, 2 hours after a lip stimulation. And then also a naive group for control, meaning those animals were also in a tank, but they were not fed any of the inedible food. So from the cerebral and buccal ganglia, we extracted RNA, and then the concentration was measured using a spectrophotometer, nanodrop. This was followed by cDNA synthesis, and then the gene expression was quantified using qPCR. And then the mRNA levels were then analyzed using the comparative CT method. So the results, these were the primers that were used for the qPCR. For GAPDH was our housekeeping gene. This is for CBP, CREB1 alpha, and CREB1 beta. Okay, so we have some preliminary results. There's still more being added to the graphs. So again, very, very much preliminary, but they do tell us a little bit of a story about what might be going on with these transcription factors. So we only have enough to say something about CREB1 alpha. CREB1 beta and CEBP are still in the works. So look forward to hearing about them soon. Anyways, so CREB1 alpha in the buccal ganglia, we can see that the naive is lower compared to the other ones, and that there is an increase in CREB1 alpha expression generally across the board. If you look here, and when you compare samples that were taken 15 minutes or 2 hours after the treatment, there is not really a significant difference between the two. If you look at long train versus short train, they're not significantly different. Same thing with long lip and short lip, not significantly different. So this shows us that the peak in expression must be earlier on because we didn't miss it when we collected the tissues 15 minutes after the treatment. And if we look here at the preliminary results from the cerebral ganglia, again, you can see the naive has much lower expression than any of the groups. And with training, there doesn't really seem to be a difference between short versus long again. However, there is the significant difference between long lip and short lip, which is interesting to note. And again, we have to add more data to see if there is something about carbon alpha in the cerebral ganglia that its gene expression peaks later on. So it could have been that we missed it when we collected tissues 15 minutes after treatment. Okay. So future work that needs to be done to better get a picture of the story behind increases or decreases in gene expression is that we need to run additional cDNA samples through qPCR. So more qPCR runs need to be done in order to allow for, like I said, a clearer vision of the changes in gene expression of these animals. And another thing to note is that we had animals that were outliers, meaning that their CT values, which shows the amount of qPCR runs that were necessary to have that peak in the amount of mRNA. So those that had very different CT values from the rest of the group, when we looked back at what the animals were like when we were training them, because we took notes, we saw that those that had, abnormally low values tended to be less interested in the training, and those that had very interesting values, either very low or very high, were those that inked during the training. So inking is a kind of defensive mechanism, and when they do that, it changes the gene expression. So it's something to consider, maybe not to take into account those animals, maybe not to include animals that didn't seem very interested in the training because they may be muddling things, making the data not so clear. So that's just something to think about. Also, something else to note is that the specific animal that we used were Plisia fasciata. Other work had been done with the Plisia californica, like in the previous research, but because of the fact that we experienced an animal shortage, we use a Plisia fasciata, which are actually nocturnal. So they don't learn as well during the day, but they were trained during the day. So it's interesting to see that there was changes in gene expression regardless of the fact that they were trained during the day, which is when they don't learn that well. So this shows that there must be still learning going on, even though it's not, they were kind of woken up at hours that they're not necessarily wanting to be so active during. Okay, so another interesting thing, like I mentioned, that there were animals that were less interested, more interested. It would be interesting to see if there is a direct correlation between gene expression and interest during training. That's some future work that we could potentially do. Thank you so much for taking the time to hear me talk. This work was sponsored by the NIH, the MARC program. Thank you so much for giving me the opportunity to do research this past summer. And like I said, this work was conducted in Bar Ilan University in Ramat Gan, Israel, in Dr. Susswein's lab. Thank you so much for listening. And if you have any questions, feel free to reach out.